So here in this rack I have the serial dilution of lambda DNA that I've prepared. So these are the dilutions of DNA with concentrations of 50 nanograms per microliters, 25 nanograms per microliters, and so on. And the first tube is just blank. As I mentioned during the lecture, blank is just a solution or the buffer as such without the DNA. So the only missing component between blank and these tubes would be DNA. So the buffer serves as blank for today's purpose. So once you've prepared the different dilutions, the next step is to transfer them to cuvettes. So these are plastic disposable cuvettes. You can see here that on one side you have a transparent glass through which light source can go and then light can be transmitted out, while the other two sides are a little translucent. So once you've taken a cuvette out, you pipette the solution into the cuvette. So once this is done, you repeat the same for all of your prepared dilutions and transfer it to separate cuvettes. And if you have to label your cuvettes, label them on the translucent side that you won't need to use. This is the spectrophotometer. The first step is to switch it on, and it's going to take a minute to warm up. So once it does, you can see that the LED here bounces off between the different modes that you can select. For the purposes of today's experiment, we are going to choose the lambda mode, wherein you can choose the number of wavelengths and the wavelength at which you want the absorbance measured at. So the first question is, it asks us the number of wavelengths you want the absorbance measured at. In our case, it's going to be 1. So just press 1 and then enter. So the next question it asks is for us to enter the exact wavelength. So 260 nanometers. And now it's ready to read the absorbance values. So this is the chamber in which you can place the cuvettes. And you can see here that there's an arrow mark here, and this indicates the direction in which your light passes through. So the light source would be on this side of the chamber, while the photodetector would be on the other side. So the arrow mark on your cuvette is on one of the transparent sides of the cuvette. So just place it in such a way that the arrow mark on the cuvette aligns with the arrow mark that indicates the direction of the light source. Once you've placed the cuvette inside the chamber, close the chamber, press read blank, and it's now setting it to 0 for this solution. So once this is done, we can move on to the next solution, which in our case is going to be DNA. So place it inside the chamber, close it, and press, once it says ready to read absorbance, press read sample. It reads 2.861. So once you're done with your first sample, take your second sample, place it inside the chamber, close, and without pressing anything else, just press read sample again. 1.513. So this is done for all of your samples. To recap, Your DNA sample is placed inside the cuvette. You set the light source to 260 nanometers. It hits your DNA sample. The transmitted light then hits the photodetector, and it then gives you the absorbance readout. So the higher the concentration of DNA, the more the light that is going to be absorbed. So lesser is the light that's transmitted, which hits the photodetector, and lesser the light that strikes the photodetector, higher the absorbance value that it reads.